Okay. Right, well, good afternoon or whatever time of day it is, wherever you are. Welcome to our third in the Vax-Hub Analytics webinar series. Just a couple of housekeeping items to start off with. So won't explain where the fire escapes are. That seems superfluous. I hope you know where your fire escapes are. We're recording this webinar, so we will try and make the recordings available to Vax-Hub members and possibly to a wider audience. Please don't let that discourage you from taking part in discussion, because if anything's raised in a discussion which you'd rather not be public in the recording, please just let Anne know and we'll seek to edit that out for you. Please also try and keep your microphones and cameras off during John's presentation, just to avoid background noise and maintain bandwidth. And if you want to ask questions, please either just type them into the chat box, or at the end when we have our question and answer session, raise your hand and Anne and I will try and moderate and bring your questions to John's attention. So if we can, without that, having got that out of the way, we can make a start. So today we have Dr. John Hickey from the University of Kansas Vaccine Analytics and Formulation Center to talk to us about the use of mass spectrometry in vaccine development. So John did his PhD in biochemistry and biophysics from Kansas in 2011, worked initially as a postdoc with Professor David Volkin and Dr. Sangeeta Joshi, which they set up the Vaccine Analytics Formulation Center. Since 2012, John is now a staff member, and he is currently a Scientific Assistant Director. And a number of you will be aware that that centre there has a number of past and ongoing collaborations in vaccine development with a number of the VaxHub partners and contributors. So John's going to talk to us about the work that they do there and the mass spectrometry analysis in vaccine development. So I think without any further comment from me, I'll just hand over to John. Stop sharing this.
Okay, everybody can see my screen?
Yes, no problem, John.
Okay. Thanks for that introduction, Steve. And I'll take questions at the end, but feel free down the line if you think of anything else to email me. I'm happy to help. So to start off, I'm just going to give you a little bit more background about our center here at the University of Kansas, and then I'll get into case studies of how we use mass spectrometry to develop vaccines. This is just kind of an overview slide of kind of our focus of our work. Mainly it's through analytical characterization and formulation development of proteins, VLPs, and viruses for pharmaceutical use. I'll get into some of these points later, but also as an academic lab, we try to publish our work and present our work as much as possible and train and employ the next generation of biopharma and vaccine scientists. So we're a team of about 22 people. We're led by David Volkin and Sangeeta Joshi. We have some permanent staff members, and they also have a number of postdocs, research associates, and graduate students. And so our collaborators and clients originate from pretty much every sector, from industry, government, universities, nonprofits, etc. And on the right here, I'm just showing our collaboration with the Bill and Melinda Gates Foundation, which has facilitated us collaborating with partners all over the world. I think about 25 to 30 partners now. So we've had this collaboration for about 5 years. And through all of these various partnerships and collaborations, we've worked with a very diverse set of biological entities, ranging from small proteins to conjugate vaccines, subunit vaccines, to viruses, both live attenuated and inactivated, VLPs, and even up live attenuated bacteria and inactivated bacteria. So with all these various biological entities with quite a diverse complexities and size ranges, really we require robust analytics to understand each molecule. And so we like to use the analogy of the elephant and blind men, where you can't just look at one aspect of the molecule, rather a comprehensive understanding of the chemical, physical, and biological characteristics of a product are required to mitigate risk and ensure a product is safe and effective. And this comprehensive understanding of a product is only achieved through sound scientific analytics. So I just have a brief slide here to show some of our analytical capabilities that encompasses both immunological assays, either plate-based or animal model-based, and in vitro through ELISAs or BLI. And then more physiochemical assays, which range structural attributes from primary to higher order structure, both static and as a function of temperature, look at aggregation propensity and even heterogeneity within the protein population. So today I'm just going to be focusing on our mass spec capabilities. Primarily we use 3 different mass spec based techniques, primarily intact mass, LC-MS peptide mapping, and hydrogen deuterium exchange mass spectrometry. So I'll have case studies in each of these 3 applications later on in the talk, but I just want to give a brief overview of kind of the role of each of these, how complex they are, and kind of what information you can get from each. On the left here, I just kind of have a crude example of how much information and how much material and time requirements are required for each technique. With intact mass, it's very quick. You can get it done within a single day, requires very little material, but also you're compromised in how much information you can get. With peptide mapping, it's a little bit more time and material intensive. Requires, it can be done within a day, but generally takes a few days, but you also get more information than intact mass. And then finally, hydrogen deuterium exchange mass spectrometry requires much more time and material, but on the flip side, you get much more structural and functional information. And so for the next few slides, I'm just going to give a quick primer for each of the 3 techniques, kind of how they work, just so everyone has a little bit of background. So for intact mass analysis, you have a protein mixture that goes through a simple chromatography. You can use this for separation, but this is more for desalting the protein mixture to get it ionized and into the mass spectrometer. And here you're looking at intact masses. I just have a figure from a paper we published a few years ago, looking at a 2 protein complex, A chain and a B chain, and we also identified a post-translational modification, a glycated B chain. And I'll actually have a case study on this later on. And then below I have kind of some product versus process related information you can get from this. As you'll note, a lot of these are semi-quantitative. In the protein mixture, you can get semi-quantitative. If you set up standard curves, you can quantify the relative abundance of these various moieties, but in general, it's a semi-quantitative technique. For LC-MS peptide mapping, again, this requires a little bit more method development where you're treating the protein mixture with a protease. You have a longer gradient to get better peak separation. In this case, you typically quantify the chromatogram using UV, either 280 to looking at tryptophans or 214 looking at the peptide backbone. And then all these peptides feed into the mass spec, which you can either look at the mass of each individual peptide, or you can fragment the peptide for identification. And in terms of process versus product related information, here this is much more quantitative. You can identify products, quantify various post-translational modifications, glycations, etc. And even if you're looking at crude material, you can quantify and identify host cell proteins. And then finally, the third mass spectrometry-based technique we use in the lab is hydrogen and deuterium exchange mass spectrometry. I'll go into a little bit more detail on this later, but basically, the way the technique works is you incubate the protein or protein mixture and deuterium, which exchanges rapidly in more flexible regions, more slowly in rigid regions, and then you use mass spectrometry to monitor this mass change as a function of time. So you can get fairly high level information from this. You can look at the inherent flexibility of the molecule, determine how protein or post-translational modifications impact both the local and global conformation, and then provide more information on protein-protein interactions, whether it's identify epitopes or looking at a protein-protein interface, things like that, or whether you're looking at protein-ligand interactions, and in this case, I just indicate you can identify active sites. So today I have 4 case studies that we've published or soon to be publishing of our mass spectrometry use throughout the vaccine research developmental cycle. And in the first case study, I will actually be referencing some of the work we did with UCL and MIT on a grant from the Bill and Melinda Gates Foundation. So this was known as the ULTRA collaboration. And the goal of this project was to develop a low-cost vaccine. In this case, the target was 15 cents a dose. And as a model system, we used a rotavirus subunit vaccine. And I should note that PATH is developing a similar vaccine in E. coli. And so we used some of their information to develop a similar vaccine in Pichia as a proof of concept. And the current vaccine is a trimer of the 3 most prevalent serotypes of the rotavirus. And it's a subunit vaccine of the surface protein conjugated to a small 20 residue peptide from tetanus toxoid that's a universal CD4 T cell epitope. And the initial workflow of this collaboration was that MIT and UCL were producing in Pichia these proteins, which are called NRV. They go through a simple 1-step purification, and then we would receive this material and then go through a quick structural functional analysis that's looking at the primary structure, conformational stability, interaction with preservatives, relative solubility, and then antigenicity. And depending on the outcome, that information is either fed back into the process to design a better molecule, or it continued on to more extensive pre-formulation characterization. So in this slide, I'm just showing our very first initial batch of NRV that was produced in Pichia. On the left, I have SDS page, and on the right, I have some intact mass. So as you can see on the gel, we have a variety of product variants. We have reducible dimers. We have glycosylated, non-glycosylated species. And then within the non-glycosylated species, we have truncations. So we have a variety of product variants. And then all these observations by SDS page were confirmed by intact mass, namely, so this is the mass we would expect for full-length protein, and instead we see a truncated species. And this truncation, if we look at the primary sequence, was confirmed to be the CD4 epitope was actually cleaved in the truncated form. And then finally, we can see high mannose glycosylation. So we undertook a variety of MS-based techniques to better understand the molecule that can help design a better one. So in this slide, I'm showing our endeavors into identifying the glycosylation sites. So we treated the sample with EndoH, which cleaves off the high mannose glycans and just leaves a single GlycNAc residue, which we can easily identify by mass spectrometry. On the right here, I'm showing the sequence coverage. As you can see, we cover the entire molecule. And out of the 4 canonical N-link glycosylation sites. We identified this +203 Dalton adduct at 3 out of the 4 sites. And so using this information, it was fed back, and the original construct was designed where 2 out of the 3 glycosylation sites were removed, and the single cysteine that caused the dimers was removed. And so through this quality by design study, we were able to show that these 3 mutations mitigated glycosylation, N-link glycosylation, and the dimer formation. However, the truncation issue was still outstanding. And so a recent paper by Lourdes from Terrett's former group at UCL tackled this issue. And I'm just going to show one slide from this paper here. Essentially, this was optimizing both the USP and DSP processes. There's quite a bit of information here, but I'm just showing a few of the aspects that Lourdes looked at for USP optimization that include growth temperature, pH fluctuations, additives, and things like that. And through this optimization, we went from 0% full-length material, so essentially we quantified full-length versus truncated material. Through this optimization, we're able to get about 60 to 75% full-length material. And then through both USP and DSP optimization, we achieved about 95% full-length material. Okay, if I can go forward. So that was it for case study number 1. For case study 2, it was a little bit more mechanistic understanding and mitigating future risk. So this was a paper we published a couple of years ago that was focused on the effect of preservatives on the stability of a monoclonal antibody. So for multi-dose formulations, preservatives are required to inhibit microbial growth during storage and administration, but are known to cause physical instability of protein-based drugs. So we studied the impact of 4 preservatives with different hydrophobicities, from the most hydrophobic methylcresol down to the least hydrophobic benzyl alcohol, and their interaction or effect on IgG1 monoclonal antibody. And then we used a variety of techniques to look at the conformational stability of the molecule, looked at aggregation propensity, and then finally backbone flexibility. So in this slide, I'm showing the conformational stability and aggregation propensity of the mAb in the 4 different preservatives. I just have representative thermograms on the left. What was interesting was for all 4 preservatives, we saw destabilization, but this destabilization followed the hydrophobicity of the preservatives. So methylcresol was the most destabilizing, and benzyl alcohol was the least destabilizing. So this was for the overall conformational stability, but also for aggregation propensity, we saw a similar trend where the most hydrophobic preservative resulted in the most loss of monomer and concurrent increase in soluble aggregates, and then the least hydrophobic preservative had the lowest effect on aggregation propensity. So we wanted to have a little bit greater detail of how these preservatives are interacting with the molecule. Is it a global interaction, or is it a local interaction that's causing this destabilization and aggregate formation? And so we use hydrogen deuterium exchange mass spectrometry. As I mentioned before, we can use this to look at flexible and rigid regions of the molecule. And actually, I'm going to go to this slide. So in this slide, it's quite a bit of data, but I'll kind of walk you through it. So essentially, we incubate the mAb with the preservative and D2O as a function of time, And then we look at the mass increase of each peptide following proteolysis. So as you can see in these profiles, we can, for some of the peptides, we really don't have a change in mass between the preservatives. But in a few cases, namely this heavy chain CH2 peptide, we see dramatic changes by mass. And similar to what I showed on the previous slide, the mass changes follow the hydrophobicity of the preservative, mainly the methylcresol showed the biggest in change, which means there's a large increase in local flexibility, and then benzyl alcohol was the lowest. So if we map all of these relative flexibilities on a homology model, which I show here, we can see that there's both global and local effects by these preservatives. So a global increase in flexibility is shown in this kind of salmon color, and then large local flexibility are shown in the yellow. So we can see for all 4 preservatives, we do have a global increase in flexibility. But what was most interesting was in the CH2 region, which is known as a aggregation hotspot, we were able to identify large increases in flexibility, and this correlated with our physical stability data. So kind of the take home from this study was the destabilization of these preservatives followed the hydrophobicity. So methylcresol was the most destabilizing versus benzyl alcohol. So this really goes to show this technique can be used to mitigate future risks. So if you were introducing a preservative for this monoclonal antibody, you would obviously not use methylcresol. Benzyl alcohol would be preferred, but also you could look at alternative preservatives that maybe don't have any effect on the molecule, at least under these conditions. Okay. Our third case study is a more prototypical formulation development and MS use in that endeavor. So this is a collaboration we have with PATH from a couple of years ago, but they were evaluating a novel mucosal adjuvant called DMLT, which was developed by John Clements at Tulane University. And currently this is being evaluated in several vaccine candidates. And on the right, I have a crystal structure of LT. So DMLT is a double mutant of this molecule. I have the 2 mutation sites here. So it's a single A chain, non-covalently linked with a pentameric B chain. So its overall mass is about 86 kilodaltons. And so part of our pre-formulation characterization, studies include looking at various degradation pathways and physiochemical stability profiles of a molecule. That includes both structural integrity, conformational stability, aggregation propensity, and finally chemical resistance or susceptibility. So I should state a little bit more about the material we see. This is produced by Walter Reed. It's from E. coli, it's lyophilized, and I have the current formulation right here. And so I won't go into a lot of the detail here. I'll mainly focus on the molecule's chemical stability. So on the right, I have just a list of common PTMs and chemical modifications that are generally experienced in biotherapeutics, 3 of which I highlight here. I'll show a little bit more detail on, and are fairly common: deamidation, oxidation, and glycation. So as part of our pre-formulation characterization, we look at the susceptibility of the molecule towards these various chemical modifications. And in this slide, we incubated the sample at 3 different pHs at 2 temperatures for a week to try to induce deamidation to see if it's susceptible, and if it is, what residues are susceptible. So I'm showing intact mass here, where we're looking at the mass of the B chain, the glycated B chain, which I'll come back to in a minute, and then the A chain. So as you can see, after a week incubation, we don't really see a change at pH 7.4 or at 8, but once we get up to pH 9, we do start to see some mass changes. And if we perform peptide mapping, we can actually identify which residues are becoming deamidated, namely asparagine 1 in the A chain was the most susceptible, followed by these 3 additional sites in the A and B chains. So in terms of glycation or chemical modifications, so we essentially performed a similar study, but instead of looking at deamidation, we looked at glycosylation. So I should note that the current formulation contains 5% lactose, which is a reducing sugar. And so after either time or temperature or pH, a typical Maillard reaction will occur where we have a non-enzymatic glycation. So this addition of 324 Daltons on the B chain. And so over time, you can see that the relative abundance of the non-glycated versus glycated species changed, namely more glycosylated species was observed over time. And then finally, through peptide mapping, we're able to identify the site of glycation, which was this lysine at residue 84 in the B chain. And then finally, in terms of oxidation susceptibility, we treated the sample with hydrogen peroxide, and then again performed intact mass and LC-MS peptide mapping. So we were able to see by intact mass oxidation shifts by 16 Daltons, which is the typical mass increase you would see with oxidation species. And then through peptide mapping, we're able to identify the methionine susceptible to oxidation. Namely, this MET 37 in the A chain was the most susceptible compared to the other MET residues in the A and B chains. So on this slide, I'm just showing our typical pre-formulation characterization and formulation studies, our typical workflow. And on the right, I kind of have a list of the various excipients we screen. So first we perform the pre-formulation characterization, identify what it's susceptible to, and then add in these various excipients to see how the molecule is either stabilized or destabilized in the presence of these various formulations. And so I won't get into too much of this. I'm just gonna kind of highlight the final part of the study was essentially we went from the current formulation to a candidate formulation, mainly through a lot of our MS-based techniques inform this decision, essentially going from lactose to sucrose. So we don't have the non-enzymatic glycation. We have the addition of methionine to mitigate any oxidation. And then a pH 7 was found to be the best pH to mitigate deamidation, but also keep the molecule in a stable conformation. Okay. So finally, case study 4. So this is mainly using MS-based techniques to identify the mechanism of a potency loss. So this is kind of a late stage development. This was a project we had a couple years ago with Protein Sciences before they were purchased by Sanofi for their influenza vaccine, known as Flublok. So it's a recombinant hemagglutinin, and it's expressed in baculovirus. I won't get into too much of this, but I want to get into a little bit more detail about their vaccine. It comprised of hemagglutinin, the full-length hemagglutinin, which comprises the ectodomain, transmembrane domain, and the cytosol ectodomain. So these natively form trimers, and then in solution, they form 30 to 40 nanometer rosettes. And so one of their typical potency assays is known as a single radioimmunodeficient assay. Essentially, it uses a agar plate containing antibodies against hemagglutinin you add in the sample, and depending on the diffusion radius, you can correlate that with the potency of the vaccine. And what they were seeing was during storage, their potency decreased significantly. So in this trace, is showing the potency over a month at 25C. We can see about a 60% decrease in potency. If we store the same vaccine at 4C, we see about a 40% decrease in potency. And then if we add in a detergent, it can rescue a little bit of the potency loss, but this is still a significant reduction. However, if we add in a mild reducing agent, known as STG, this does help mitigate some of this potency loss. And what was interesting by STS page, even at T0, we can see this laddering effect. And then under these mild reducing conditions with STG, this laddering effect goes away. And we're primarily observing monomers, dimers, and a few higher order species. And then we also looked at the free thiol assays because we knew from the STS page that disulfides were playing a role somehow in this potency loss. And as we observe over the course of a month under the same conditions that they observed this potency loss, we can see a dramatic decrease in the availability of free cysteines in the molecule. So this really indicated that the cysteines are playing this role. But then we want to know which cysteines are involved. Is it in this ectodomain? Is it in the transmembrane cytosolic domain, etc.? So we developed a differential alkylation assay, essentially where we denature the molecule with guanidine, add in a reducing agent. So this will only alkylate free cysteines. We go through a buffer exchange, reduce it, and then we add in a second alkylating agent, iodoacetamide, and perform proteolysis and peptide mapping. So if we look at the relative abundance of NEM versus IM, we can really monitor the redox state of each cysteine over time or under different stress conditions. And so that's what we did. We essentially set up another 1 month incubation and quantified the relative abundance of each labeled cysteine. So I just have representative spectra on the left showing the ion chromatograms of iodoacetamide labeled cysteine versus NEM labeled cysteine, and we quantified the relative abundance of these two peaks over time. And what we observed was for under conditions where it's just under temperature stress over a month, we saw a significant decrease in the relative abundance of the free cysteine labeled, or free cysteines, I should say, over time. And this all correlated with the SHRID potency assay. And I should note that really in the in the ectodomain, we really didn't see a change in the relative redox states of the cysteines. It was primarily comprised to the transmembrane and cytosolic domains. And so our hypothesis for this in vitro potency loss was that these rosettes initially in the SHRID potency assay are treated with a detergent. These rosettes get dispersed and you have an active potency reading. However, over time, these disulfides form the cytosolic and transmembrane domains, which prevents the dispersion from the detergent. And so it appears that the SHRID is inactive. And so subsequent studies by Protein Science, where they mutated various cysteines in the transmembrane and cytosolic domains, were able to show that if you substitute these cysteines to serines, you can essentially abolish this SHRID potency loss. So that was fairly quick. So I left some time for discussions and questions. So I want to thank everybody for their time. And if you have any questions, please let me know.
Thank you, John. That was a nice description of the versatility of mass spec, I think, in terms of how it can contribute to pretty much all stages of vaccine development, from clone selection all the way up to formulation and stability assays. So, are there any questions that anyone would like to raise? Either raise your hand and we'll get to you, or type something into the chat box.
Wait.
So, Adil, you have your hand raised, I see.
Hi, Steve. Hi, John. Thanks a lot for that great talk. While other people gather their thoughts, you know, I always have a million questions. So some are good and some are mostly bad. But John, it sort of occurs to me, sort of mass spec. It sounds fantastic, but you had pretty clean and beautiful looking preparations of proteins. So, you know, in terms of, you know, you kind of talked about the different methods and the amounts of protein needed, but maybe could you sort of comment on, you know, purity and quality of preps?
Yes. So we are a little bit lucky in that we don't do any production here. We get material from our collaborators and clients, which is typically quite pure material. So it makes performing these assays fairly easy. But I showed a little bit of this earlier with our collaboration with MIT and UCL, where even the picky material is still, let's say, 80 to 90% that's coming out of the cells, so that's still quite pure. It is challenging the more complex, obviously, the sample is. But I would say probably the more challenging part is actually not the proteins, but more of what else is in there. So detergents are a big hindrance towards any MS-based techniques, and depending on whether they're, you know, small molecule additives are in there, can really affect what you can do with it in terms of MS-based techniques.
Yeah, thanks. I think that's really a key point. Is if you're trying to, you know, if you don't, firstly you need quite decent amounts of purity, I think, and maybe the quantity is not not the limiting factor. It's absolutely the excipients, as you say. So I mean, the VLPs that you were talking about, these are non-enveloped VLPs, right?
Correct. Yes. Yep.
So, I mean, do you foresee like problems with, say, the enveloped VLPs, so the membrane associated virus-like particles? Do you think there's any special considerations?
Yes, so those are always a challenge. Any kind of transmembrane or encapsulated protein-based drugs are a big challenge, because one is just from a chromatography perspective, getting them bound and then unbound to a column is a challenge, and then even more so getting them ionized and into the mass spec is very challenging. Typically you have to do some tricks to get them into a gas state, either derivatize them or maybe if you're performing proteolysis, make it in such a way that you have some of the surface exposed, more MS-friendly parts of the molecule. But yes, that is one of the big challenges. And you see this a lot in the proteomics field, where soluble proteins are relatively easy to look at, but really the membrane-bound proteins are a big challenge.
Yeah, thanks for that. I just make a comment here. Some of the stuff that we were feeding into John and his guys from the Ultra project, that was literally pretty much straight out the fermenter. It was a secreted system, of course, so it is, but it wasn't pure by a long way. So John maybe been a bit polite, shall we say. John, if no one else has got a question just coming up, I just wanted to comment at all about sort of the uses of, I see a lot lately about sort of people talking about using mass spec for HCP characterization and level detection. It's not commonly the most used method at the moment, but it seems to be something which there's a lot of attention towards. I just wonder if you had any thoughts on where that's going and how feasible that really is.
Yeah, so I would say it's absolutely feasible. I mean, these days, mass spectrometry is fairly straightforward, especially with established systems, whether it be like Pichia or E. coli, where your HCPs are fairly well understood. So as long as you have a decent idea of what's there, you should be able to identify them pretty easily and be relatively confident in your ability to quantify them. Yeah, I would say, I mean, it just obviously requires some development to better understand kind of what's there. But once you've established that kind of baseline, it should be fairly straightforward to quantitate.
Okay, thanks. Anyone else? Adel, I can see you there again.
So Steve, I have a question for you actually. So how much do you think kind of in the processing field do you think is incorporated for any MS-based techniques, or is it primarily for like gels and those kinds of things?
Well, I think, a lot of the processing field is relying on gels and so forth. But in a way, our collaboration with the Ultra is an example of the limitations of that. Whereas we had that epitope at the terminus, and for function, as far as we know, that epitope is essential, and we were getting it was getting processed off. Yeah, and you would only see that on the gel when you'd lost the entire epitope, the whole 14 amino acids. But you were telling us, when we'd lost a couple of amino acids, and so if we hadn't had your mass spec analysis in that, we would have been saying things were good quality which weren't. Yeah, I guess is the outcome in there. And I'm wondering how often other people are saying that, looking at a gel and saying yes, that's fine, and in reality it's not, and how much that contributes to studies not being as active as people expect them to be. So I think I've been very much convinced, that it has a lot of mass spec has a lot of contribution to determining product quality, even at very early stages.
Yeah, I was going to say we actually typically run into that, especially with our collaborators who are kind of working with early material where they think they have this highly pure, homogeneous mixture, but when we perform our MS-based techniques, we have to let them know that, you have this post-translation modification or you have this truncation, and you have all these kind of quality issues you need to deal with.
Yeah, and because we were feeding that into the early stages of our project, then we were able to deal with that. So so I think the sooner your sort of assays feed in, the better. Yeah. Anyway, Adel's waving.
I mean, actually, that's a really interesting point you guys are making. It's just in terms of feasibility. So, I mean, I guess for Luddites like myself, the accessibility or even thinking about what mass spec can deliver is sometimes daunting. But you made it very clear, John, not to this. I thought it was a very astute kind of presentation of the usefulness of mass spec. But if people like myself are thinking to design projects from scratch, how could we best? What do you think our workflow should look like in order to kind of maximize on its application So I'm interested in virus-like particles. I wish to make a new virus-like particle in Pichia or another yeast. And so what are the considerations I should sort of build into my design? I know this is a really broad question, but but it's, are there things that you think, because it sounded like, you used with the sort of the VLP you were discussing with Steve, is, it had multiple components and it was post-translationally modified, and it had, certain it was secreted, so it was processed through the yeast. But if we're looking at, say, intracellular expressed, stuff that's not secreted, what are the tips or tricks you could kind of, or do you have any such information?
Yes, I would say it all comes back to, well, if you have a potency assay, I mean, that trumps everything else, because if you have, let's say, all these modifications or truncations, but it has absolutely no effect on the potency, then really, how much do you really need to worry about it? But if these modifications do have a detrimental effect on potency, then obviously you need to mitigate those modifications. So I would say for the mass spec side of things, it's really should be viewed as a tool to help inform the process and product. And then depending on the effects of the process, you can either use it heavily, like we did, like Steve and I just talked about, kind of to inform both designing a better molecule and a better process, or just simply as kind of making sure everything's okay after purification. I think I answered your question.
Yeah, no, you did. I mean, that's really, yeah, I think that's key. So potency for us might be, say, particular antigenicity, for example, sort of conformational epitopes.
Yep.
So, sorry, my kid is in the background playing Minecraft. I'm sorry.
Nice.
He's just been hacked, apparently. No, thanks a lot. Thanks a lot, John. Okay, I'll mute my microphone now. Maybe we can edit this part out of the.
Okay. Anything else for anyone? Comments, questions? No, I guess not. I guess hopefully you've convinced everyone that this is something they should be looking at.
Yes, and I'm happy to take any questions offline if anybody has anything.
Yeah, I'll definitely send some emails. Thanks a lot.
Sounds good.
Okay, well, in that case, I guess if no one else has anything else to raise, we can wrap it up. Thank you, John, very much. As I say, there will be hopefully a recorded posted up of this session, which we'll send you links to. There is a plan still to try and continue these VaxHub webinars, even though as people sort of move back into the labs and get access and so forth. We'll try and come up with a format. Oh, hang on, wait a minute. Marco has a question, which says, can these methods be used in continuous modes, i.e. coupled to reactor outlets?
I think theoretically, yes. You would have to be careful of kind of what's in the reactor, as I was saying earlier, in terms of small molecules and their effect on the LCMS, but I think in theory, yes. It probably would be easier to take samples rather than some type of continuous to kind of spot check throughout the process rather than a continuous kind of workflow.
Okay. He's asking us for post-processing steps for mRNA. It's obviously what he's thinking.
Processing steps for mRNA. That would be a challenge. Trying to think of how that would be done. Yeah, I think that would be very hard to establish, and especially in a type of a continuous workflow. That'd be very challenging.
Just along that point, actually, I'm so used to thinking of proteins all the time. Is the mass spec used for RNA, DNA work?
Not too much. They're typically poor ionizers, so getting them into the mass spec is a challenge. For like glycans and those kinds of chemistries, it's widely used, but for DNA, RNA-based molecules, it's quite a challenge. You typically have to derivatize them and then do some other manipulations to really get them in the mass spec.
Okay. So I think the answer, Marco. Sorry.
Do the timelines allow it? So I mean, if you're trying to do an in-process sort of, I mean, if it takes a few hours to get the answer, does it in terms of if it was proteinaceous and compatible with the mass spec, do the timelines permit it to?
Oh yeah. So I mean, typically for intact mass, I mean, running through a LC and performing all mass spec can be done in a matter of minutes. It's just kind of what information you want to get out of it. If it's a complex system, you may need to expand that window to have better separation, or if it's a fairly simple system, then you can do it continuously.
Okay. Anyone else sneaking something in? If not, what was I saying? I was saying that we're planning to try and maintain these webinar series, and we'll keep you updated on that. Anne, is there anything else I need to inform people about?
No, I don't think so. Not for the moment. Thanks a lot, Steve and John, for hosting and accepted to be the speaker of today. So thanks a lot, guys, for doing that today.
Okay, so thank you, everyone. Stay well, and see you soon, hopefully.
Thanks. Bye.
Bye.